Okay, our next speaker is Dr. Yael Ottolenghi from Sheba Medical Center, will tell us about the Sheba Pandemic Preparedness Research Center and what it does, I guess, to prepare us for future pandemics. But let's see.
So hello everybody, thank you Itai for the introduction. So my name is Yael Ottolenghi. I'm the representative of SPRI, the Sheba Pandemic Preparedness Research Institute, which I will tell you about in a minute. Just a comment that I'm really excited to be here today. I'm a former student here and I was a researcher for a long time with Professor Johnny Gershoni, and I'm sitting in front of my mentors, Professor Johnny Gershoni, Professor Gili Ragev Yochai, Dr. Danny Duek, who was a visitor here, and in front of my best friends. So it's really challenging, and so forgive me for my stuttered English, and let's go to business. So, navigating future pandemics with enhanced preparedness. How do we do that? So our story began in January 2020 when COVID-19 pandemic has started to spread, and back then the Sheba Prevention and Control Unit, Infection Prevention and Control Unit, headed by Professor Gili Ragev Yochai, has started a recruitment, had led a recruitment of over 9,000 healthcare workers. The recruitment has started with the first vaccination that arrived to Israel in aim to run a long-term study that still is, in order to provide insight into the real-world effectiveness, vaccine effectiveness, and to understand the immune response towards SARS-CoV-2. So in order to do an extensive and comprehensive study, you need a methodology in order to collect data. So the methodology for collection data was as follows. All the vaccination data and the PCR results and the antigen results were collected from the healthcare workers, of course. A detection of symptomatic cases during the first year or a bit more. Every day the healthcare workers of Sheba received an SMS message in order to ask them if them or their family members are feeling okay. If so, if they didn't and they feel like influenza-like symptoms, they have been asked to be tested. In addition, a computer-based tracing system was built quite quickly in order to detect exposures and asymptomatic cases. So in case there was a positive PCR or antigen test, immediately they were monitored, and then the Infection Prevention and Control Unit tracked them in order to participate in these healthcare workers. Of course, comorbidities and demographic data was collected, age, sex, background comorbidities, and of course, blood samples were collected. The healthcare workers were given blood every month, and they have a notification to go back and give blood every month. And subgroup of, from this cohort, also we took some PBMCs from them, immune cells. So by this methodology, an enormous one-of-a-kind collection of data was collected, and a repository of samples that we longitudinally track about these 9,000 healthcare workers. So following these extensive and comprehensive studies, this is the key achievements that I will go over them briefly. Vaccine effectiveness, correlate of protection, immune response, reactogenicity and uptake, clinical trials that were done in Sheba, and vulnerable population that was assessed. So all these key achievements were published in above 60 papers in renowned journals like Lancet, and New England of Medicine, and Nature, etc. And the output of these studies also has an impact on worldwide and national decision making. For example, giving the administration of the third vaccine dose. So following our understanding, after the COVID-19 pandemic, we understand that we are not prepared for the next pandemic. So how do we prepare for the next one? So what are the steps? So many strategies are established and be launched or are being discussed. The main of them is the pathogen surveillance. However, this strategy, the pathogen surveillance, does not address immunological hidden variables. We cannot rates of asymptomatic infection cannot be detected, population-wide disease susceptibility, cross-reactive immunity dynamics cannot be assessed. However, what can we do? So we can discover monoclonal antibodies or rediscover them. We can design immunogens, and we can think about boosting strategies that will be better. And we can targeted detection and surveillance of strategies. So 3 years ago, Dr. Danny Dweck, who is a senior investigator and chief of the human immunology section at the VRC, and he's the entrepreneur and the director of the PREMIS program. So what is the PREMIS program? PREMIS is a pathogen agonist anticipatory reagent and countermeasure discovery program led by the VRC. Its aim is to establish a preemptive repository of immunobiological countermeasure, immune assay, pathogen-specific knowledge to accelerate the response to potential pandemic threats. And the process is to immune analysis of human cohorts to detect reactivity against potential pandemic viruses. So by this aims and process, the deliverables can be reagents and data resources for early detection and diagnostic assays, monoclonal antibodies with potential for prevention and therapy, and immunogens suitable for vaccine development. So this is the workflow that PREMIS is following. Look at the map of the world, track the places that there are viral threats, but whether it is a specific country or a broad area like the Middle East, for example. Take from each location serum samples and immune cells. Study the virus, define the viruses, discover them, and select the one that are threatening us. Then design the antigens. Use the serum samples in order to do a serological test and to analyze this antigen, and after validate them, you can use them to identify antigen-specific B cells. These you can be sorted and you can deliver monoclonal antibodies. These monoclonal antibodies can be functional, they can be further characterized, look for the best one, and at the end of it, we have reagents and data for early detection and diagnostic assay. We will have immunogen suitable for vaccine development, and we have monoclonal antibody therapeutic potential. So in addition to all of this, this procedure from sorting the cells, the moment you have the antigen design, to a functional analysis of hundreds of monoclonal antibodies, Dr. Danny Dweck has designed and developed a new methodology for a single cell sorting, which I will explain about in a minute. So this technology enabled to do it very rapidly. So in 7 days, you have hundreds of candidates of monoclonal antibodies that can be potential reagents for therapeutics. So the PREMIS program has mapped the viral threats. This is the global map. You can see in red the newly emerging viruses, and the blue one are the re-emerging viruses. And the concept is to have a partnership. The PREMIS program wants a partnership in every spot there is a virological threat, in order that you have a local capability to accelerate countermeasures development. These places, these stations are collecting the serum samples and the immune cells, taking them, transferring them to the NIH, to the VRC, where the development of the countermeasures is being done. So as you can see here, we have also SPRI, the Sheba, and Israel. Israel is also part of the PREMIS program. However, it is a different entity. It's an independent entity. As I showed you before, the comprehensive study of the SARS-CoV-2, the Sheba healthcare workers, and we intend to do our own research and development here in Israel. Therefore, Sheba recognized also the need to be more prepared for the next pandemic, and therefore the SPRI Institute was established. The Sheba Pandemic Preparedness Research Institute was established 1.5 years ago. So this is the core team of SPRI. Professor Gili Regev Yochai is the director, the senior scientist advisor, Dr. Yael Ottolenghi from the VRC, the chief virologist is Dr. Yaniv Lustig from the Ministry of Health from the Central Virology Lab, chief epidemiologist is Noam Barda, clinical and epidemiological research manager, Dr. Gili Yosef, and myself, I am the head of the SPRI laboratory. So what are the aims of SPRI? So our aim is establishing a multidisciplinary, multifaceted, and collaborative research project to translate clinical expertise into products for medical use, diagnostic, therapeutics, and vaccines, and deploying these biological countermeasures promptly in the event of epidemic and pandemic threat. So in order to accomplish our aims, and as a part of effort to turn research discoveries into life-saving solutions, we need the implementation of new technologies. So as part of the PREMIS program and our intense collaboration with Dr. Danny Dueck lab, we're implementing the newly technology for monoclonal antibody discovery. So I will go over it briefly. The technology is termed RATPIG, Rapid Assembly, Transfection, and Production of Immunoglobulin. So the concept is take the PBMCs, the immune cells, peripheral blood mononuclear cells from a person, and stain them with specific antigen that you are desired to look for monoclonal antibodies. For example, if we are looking for broadly cross-neutralizing antibody towards SARS-CoV-2, so take the RBD, the JN1, this is the current variant, and stain the cells. We are looking at the memory B cells, the B cells that have the antibody which is anchored to the membrane and the DNA of the clone itself. So we are sorting the cells, single cell sorting towards these antigens, and each cell is going to individual well. So we have 96 well plate. In each well we have one cell memory B cell. This plate is then going to RNA purification and cDNA synthesis. Afterwards there is enrichment of the DNA of the heavy chain and the light chain. These genes are going to be cloned and expressed in a human XP293 cells. So eventually we have mini culture. In each well we have a mini culture of the same memory B cell that was in the well. In parallel we are sequencing each cell, so we have the paired genes of the heavy chain and the light chain in the same well, and eventually we have hundreds of monoclonal antibodies, and we can further characterizing them for binding, neutralization, and the best one of them are being the concept is to take them, the best one of them, and to go to clinical trials and mass production to have a therapeutics. In parallel to all of this, we can sometimes do the 10x genomics. This is a technology that enables repertoire, the understanding of the repertoire of memory B cells. So by sorting both for 10x and for RATPIG, we can have a very comprehensive understanding, try to understand the immune response. As I told you before, this technology is rapid and robust. From day of sorting to the map production and have hundreds of candidates, it takes 7 days. And this is in contrary to traditional sequencing, cloning, and expression of monoclonal antibodies that usually take something like 3 months. So now I want to share with you the first collaboration with the NIH and the first discovery of monoclonal antibodies, which are SPRI antibodies. So although we are in a post-COVID era, an ongoing wave of infection of SARS-CoV-2 are still here, and we have vulnerable population like elderly and immunocompromised, and there is an urgent need for therapeutics against SARS-CoV-2. Therefore, the combination of the Sheba healthcare workers that I talked before and implementation of the novel technology that I just encountered with will enable to us to discover monoclonal antibodies towards SARS-CoV-2. So this project was handled by Dr. Yaniv Lustig, who is the head of the Center of Virology Lab of the Ministry of Health. I have to say that this project was started when SPRI was a fetus. It was back then, but now we are continuing to do this study. So Yaniv was in sabbatical in Dr. Danny Dueck lab and wanted to discover broadly cross-neutralizing antibodies and to understand the immune response following Omicron infection. So he used the Sheba healthcare workers and looked for the differentiate memory B cell response following Omicron infection in naive, third and fourth dose SARS-CoV-2 messenger RNA vaccine recipient. So Yaniv implemented the rapid technology, and by doing it, he generated 2,500 monoclonal antibodies from 26 samples. And by doing in parallel the 10x genomics, and the RATPIG, he sequenced almost 15,000 sequences of monoclonal antibody towards the Omicron, the BA1 variant. So I will just go over briefly the results. This is not published yet, and we are still going in characterization of these antibodies, but this is the neutralization analysis after we generated these monoclonal antibodies, test their binding to BA1, and this is the neutralization analysis of the binders. Here is a panel of SARS-CoV-2 variants, and I will pay your attention to this graph. You can see it here, and the underlined group, which is the primary infected individuals who met the B1 variant for the first time, as opposed to all the rest of the groups, the 6 rest of the groups that were vaccinated with the ancestral virus for 3 or 4 times. So as you can see here, by neutralizing for the neutralization of the wild type variant, you can see that the one that were primarily infected with Omicron, they cannot neutralize, they have a lower neutralization of the wild type as opposed to the vaccinated individuals that encounter for many times this variant. The Delta, which is also primary variant, also the picture is the same. And if you look at the Omicron variant, this is the BA1, for example, you can see that the primary infected individuals neutralize it very well. And also the BA5 variant, which is also an Omicron, so the neutralization is getting higher. And I want to take your attention that if we look for a neutralization to BQ1 and XBB, which are later variants, we can see also neutralization even though these individuals were not met or encountered these variants. So our immune response has the potential, of course, to look for, because of the cross-reactivity and similarity, has the potential to have a more broadly cross-neutralizing antibodies, even though we didn't encounter the specific variant, as well as SARS-CoV-1, which was in 2002, so it's the same picture. When we look at the sequencing, and here is look at the VH usage and the somatic hypermutations. Concerning the VH usage, we didn't find any significant outputs from this sequencing, but if we look at the somatic hypermutation and we divided these 26 samples into the primary infected, which are in blue, and the red group is vaccinated but were not infected with Omicron, and the vaccinated which were infected with Omicron, you can see the same picture both for the binding and for the neutralization monoclonal antibodies, that the level of somatic hypermutation in the primary infected individual was lower, and this is quite reasonable because they encountered the virus only once, as opposed to the vaccinated and infected individuals that encountered the virus 3, 4, even 5 times. So the immune response has a longer time to stimulate. And as I told you, our mission was to find a broadly cross-neutralizing or highly neutralizing antibodies. So eventually we focused on 2, SPRI1 and SPRI2, and we test them for an expanded panel of variants. And if you can see here in the green frame, so SPRI2 is unable to neutralize all the primary variants, as opposed to SPRI1, which cannot neutralize none of them. If you look at the Omicron variant, and the BA1 is here, of course, the first one, you can see that SPRI1 is specifically neutralizing the Omicron variant, and SPRI2 as well. And if we look at the JN1 subvariant, which is the present variant that is around us, so you can see the SPRI2 is also able to neutralizing it, and SPRI1 does not. So if I will just summarize the characterization of these 2 antibodies that we are going and trying to understand furthermore. So SPRI1 was derived from primary Omicron infected individual, the one that were encountered the virus only once. So it is very specific to Omicron. This is the VG family, the IGHV434, and by competition ELISA, he was classified as class 2. SPRI2 was derived from 4th vaccine dose recipient, and he broadly cross-neutralized all the variants that we tested, and it was the VGene family is IGH353, which is quite common, and a lot of monoclonal antibodies that were discovered in the NIH are quite similar, and it was classified as class 1. So this was the first study, and we intend, of course, this was an opportunity to test our capabilities. And we have the Sheba healthcare workers, we have implemented technology, and now in order to be more prepared for the next pandemic, we need to be monitored immune threats. So for that, SPRI has recruited diverse cohorts in order to explore the immune response and look for these immune threats. So we'll go over the cohorts we have. So the Sheba healthcare workers, this is the cohort that you are familiar with from the beginning of my talk. The West Nile virus. West Nile virus is a disease that 11% of the population is usually infected with it. We don't know because it's an asymptomatic disease. However, for elderly and immunocompromised, this is a problematic and risky disease, and we don't have any therapeutics. Therefore, we took the Sheba healthcare workers that we have and looked for the positive West Nile virus people. And from there, we further test the neutralization and ask for the PBMCs. And in order to accelerate the process of discovery of monoclonal antibodies, these PBMCs were sent to the NIH and in order to find neutralizing antibodies as therapeutics. And our honored guest, Danny Duek, who just arrived a week ago, they did the RATPIG methodology, and he told us that they found 12 monoclonal antibodies that can be candidates for therapeutics. So this is really exciting, and we hope that in a year we can do a clinical trials and test these. Another group is the travelers. These are individuals that were infected with dengue. There are 4 serotypes of dengue. Here we are more concentrated on diagnostic and less on therapeutics. The Hajj pilgrim. This is an interesting group because here we don't know exactly what is the pathogen. Hajj is an event when millions of people are gathered together and then going back to their homeland. So this is a great opportunity to spread virus and pandemics. So here we recruited a cohort which were going to the Hajj from the West Bank. They go to the Hajj, and then we collected samples before and after their journey. And the one that were complained on sickness, we tested their samples, look for the pathogen that caused their sickness. So we are still trying to identify the pathogen, but we are doing a shotgun analysis and binding analysis towards multiplexing binding of panels in order to identify what is the cause for the pathogen, for the infection, sorry. And another group is the zoonotic exposure, the bird flu. This is over the years there were some bird flu outbreaks. In 2020 there was a major severe outbreak in the north, in the Ahula resort, and a lot of cranes, agurim, were dead, thousands of them. And even though, even here 4 months ago, there was an additional outbreak, even though it was less severe. So our concept is to recruit people that were involved or were exposed to these dead birds, veterinarians, people from the national park authorities, people that were take out the dead bodies of the birds and were exposed to might be exposed to the virus. So we took the PBMCs and in parallel we are synthesizing the antigen, the H5 and N1, look for the sequences, the following outbreak that were before and this year, in order to have antigens to sort the cells to find monoclonal antibodies. Hantavirus, this is a new cohort. It's collaboration with Dr. Danny Duek lab. Hantavirus, we not know about it a lot from here, about the hantavirus here in Israel because not so studied, but it's a major problem in the States and the South America. And the last cohort is the hospitalized aseptic patients. This is a major problem of antibiotics microbial resistant patients that are infected with Acinetobacter, and if we will use monoclonal antibodies as an alternative to therapeutics, it could be very helpful and might be a good solution for them. So these were our cohorts, and we are still on guard and to be, if there are more cohorts that need to be recruited and to discover. So I told you about the Sheba healthcare workers as the first pillar of SPRI, from where everything was germinated. And the technologies and the monoclonal antibody discovery technology that we are implementing in our lab, and the cohorts, which is the third pillar of SPRI, and hopefully by merging all of this, so research and development at SPRI will pave the way for enhanced preparedness in navigating future pandemics. I would like to thank a lot of people that are involved in this institution. The SPRI core team, Professor Gil Regev Yochai, Dr. Danny Duek, Abe Mittelman, Yaniv Lustig, Noam, Gili Yosef, my lab members. Forgive me for not counting everybody, but I just mentioned the IPC unit, the infectious disease unit, the microbiology lab, the National Virology Laboratory of the Ministry of Health that we are working with them. We synergistic collaboration for a long time. The NIH, of course, for Danny Duek and his lab members that are mentoring us with professionalism and generosity. So thank you. The Sheba Central Lab and the Sheba Management, Professor Price, Professor Haratz, and Dr. Dominicini, which are encouraging us and supporting us. and for the Ministry of Health and the Hemsley Charitable Trust for their kind donation. And many thanks for you for listening. And just if you find exciting about what I told you before, so we are recruiting people to our lab. So if you are interested, feel free to contact me. So thank you again, and I'm willing to answer your questions.
Thank you, Yael. I must say this is a huge endeavor. So is the funding from the Ministry of Health and the Health Fund sufficient to keep it going for the long run?
This is an interesting question. Indeed, this procedure is very costly, and we have, I think we will manage, if we will find, if the research will be fruitful, we'll be able to, for the long run, we will have the ability to stand here and develop countermeasures that will be helping us to keep hanging here.
Okay, questions from the audience? Natalie, human beast sorting.
What is the efficiency of the process?
Actually, I have a...
Thank you.
No, don't go. I have another issue to discuss. So for your dengue program, I challenge you to isolate antibodies that will not induce antibody-induced severe dengue. So because there is this issue about primary exposure to dengue that puts you at risk when you're re-exposed. But perhaps antibodies can be discovered that will nullify the spread.
So first of all, there is a vaccine for dengue, and
am I talking nonsense, Johnny?
No more than usual.
So first of all, there is a vaccine for dengue, and it's quite useful. And dengue monoclonal antibodies were already discovered by the Red Pig platform, by the Premise program. So this is not our effort to deal with this, and they are useful. Yeah.
Okay, thank you very much.
Thank you. Now I can go.